Hello, my name is Mike Ward. I'm the head of content at Emalfarb Intelligence Insights. That's script, pink sheet, in vivo, and generics bulletin. We're here at the Biotech Showcase in San Francisco. It's the start of 2019, and it's a time when companies get together, investors get together to sort of review what's been going on in 2018, but also look forward to the prospects for the industry in 2019, and also some of the challenges. Clearly, one of the challenges that faces the industry is the cost of pharmaceuticals, and one of the aspects of that cost is, in fact, how much it actually costs to produce these new biologics. I'm joined by Mark Emalfarb, who's the CEO of Dyadic, which is you're sort of a process development company.
We're an enabler of biopharmaceuticals to help speed up the development and lower the cost.
Yeah. So most of the biologics these days are produced using either, what I think you'd call suboptimal systems. So could you sort of just first describe what you sort of see the problem is, why the existing processes are suboptimal?
Right, so our belief, and I think the belief of many growing people in the biopharmaceutical industry, is the cell lines used to produce biologics are inefficient and they're slow and they're expensive. So the media cost to feed like a CHO cell, Chinese hamster ovary, is very expensive. It was developed over the last 20, 30 years to push a cell line to do things it wasn't naturally made to do, where our technology runs on glucose, very inexpensive media, at much higher levels. So like 14 grams per liter in 7 days versus 2 to 4 grams per liter in 14 days is an average of an industry at 1/20th the media cost. And then when you get done in using a CHO cell, they're using, they have to do 2 viral inactivation steps to kind of purify viruses away. And we don't have viruses in the first place, so you save money on the downstream by not having to do those processes.
So your approach is in fact to use fungi.
Right, a unique fungi.
Yeah.
So not all fungi are created equal.
Right.
And so we've developed over the last 2.5 decades a fungal expression system to make enzymes at massive scale, like 500,000 liters, at 120 to 150 grams per liter of total protein. We've made an enzyme at 80 grams per liter of a single protein in the past in the industrial space, and then we sold that to DuPont for $75 million a couple years ago, and we kept the rights to that cell for pharmaceuticals, things like antibodies, antibody fragments, Fc fusions, vaccines, antigens, virus-like particles. So it's a platform. And we're using that platform and refining it to make human-like proteins at extremely high levels at low cost, faster, to break the cost barrier.
So the DuPont is using the technology to do what?
So BASF was one of our licensees, Shell, DuPont purchased it for the industrial, to make enzymes for animal feed, for biofuel, for industrial applications. And so we're taking the power of industrial biotech and bringing it to the pharmaceutical industry, where we had to make things for like $1 a kilo at 500,000 liter scale. Here they're making things at $1,000 a kilo at 2,000 liter scale. So we're bringing in modern biology, synthetic biology, and applying it to a hyper-productive cell line and taking advantage of Moore's law in biomanufacturing.
Right. And in terms of actually sort of developing it, do you have pharmaceutical partners that are helping you sort of, bring this forward?
Yeah, so we've, in September of 2018, looking back and then we'll look forward, we signed a deal with Sanofi Aventis for multiple proteins against multiple classes of drugs, from vaccines, the antigens, the antibodies, et cetera. And they're funding that fully funded research. We have a project that's ongoing with Mitsubishi Tanabe Pharma. We're working with the Israeli Institute of Biological Research on epidemics, pandemics, things that they're interested in there. We're working with the European Union in a program called ZAPI, Z-A-P-I, and that's a quick access vaccine program. In fact, in there we've hit baculovirus and E. coli, which are systems that they're using, and C1, our technology. The difference is they're making less than 10 milligrams per liter. we made 723 milligrams per liter on that particular antigen. So you're talking about their target was 100, and we're already 7 times that level, and we can push it higher, but there's really no need.
Right. So what are the sort of both I guess the sort of the technical challenges that you still face, but also I guess the sort of the commercial challenges as well?
Yeah, so let's address the commercial ones first. First of all, we believe all lives matter, not just some lives. And the pharmaceutical companies in general are more focused on 10 years out, new drugs that they can make a 90% margin. And the existing drugs, nobody's really paying attention to reducing the cost other than biosimilars. And a biosimilar is made in the same way. So if you have an inefficient cell in the first place, yes, you're going to be able to reduce the cost somewhat, but you're not really taking it to the level that we need to really address the problem. And as FDA Commissioner Scott Gottlieb mentioned, I think it was in August last year, 2% of the American public gets access to biologics, 40% of the federal budget for prescription drugs. That's insane. So people aren't getting medicines. So we want to work with pharma companies, biotech companies, the governments, about helping create a new cell line, C1, not new actually, it's 2.5 decades old, but new application of it to bring the power of industrial biotech to pharma. So one of the things we'd like to do is work with the FDA, for example, orphan drugs. They have status quicker reviews. We think they should have orphan status for new cell lines that can actually bring drugs to more people. So if you have an orphan drug that's say 1,000 population, but you have 10 million patients that aren't getting drugs, or 100 million, maybe you ought to get the same status.
So to be successful, who's your biggest target to convince? Is it companies who might have to re-engineer their processes, or is it the regulators who could possibly see the market access opportunities?
Well, it's both for that, but we're working with obviously the companies, big pharma, top 10, top 20, working with small biotechs that are expressing genes and having problems getting them moving into the clinic, let alone commercially viable. A lot of these new molecules, the complex things like bispecifics, trispecifics, are more difficult to make. The expression levels are low, and we're able to get some of those programs back into the clinic. So the regulatory agencies are just starting to look at new ways to do things better, quicker, and cheaper. So it's both.
So okay, and sort of going forward, I mean, sort of you mentioned that obviously you're now doing some deals. Is momentum picking up? I mean, you're at a meeting like this, right. JP Morgan, Biotech Showcase, where the whole industry is here. Right. What sort of traction are you able to get?
Well, last year we did 8 deals in last year, and this year we believe we'll do more than 8 deals in the first quarter or second quarter combined. But the traction is getting bigger, quicker, and cheaper, and going to higher levels. Because in the last 2 years, when we made this transition from industrial biotech to pharma, we've created data that's showing record levels of productivity that are just getting started. So even though as good as the numbers are now, they're going to get a lot better with a little more effort.
Okay, great. Well, Mark, thanks very much for sharing the story with us, and good luck.
Yeah, thank you very much.
To us all.
Nice to see you. Yeah, well, it's all about bringing affordable healthcare to people, and that's what we're trying to do. All of us. Thanks.